Dr. Thomas Seyfried, welcome to Bendiksen Airways.
Yeah, well, thank you very much, Alexander. It's nice to be here.
So how would you introduce yourself?
I'm Professor Seyfried, professor of biology at Boston College here in sunny Chestnut Hill, Massachusetts.
The reason I wanted to speak to you is because I came across your theory about cancer being a metabolic disease. Could you tell us how you developed this theory?
Well, the theory comes from the work of Otto Warburg, the famous German scientist from the last century. He originally proposed that all cancer was a disorder of mitochondrial energy metabolism, forcing cells to generate energy through fermentation or energy without oxygen, basically. And his work has been widely recognized. I looked at what he said. At first, I had no clue what he was doing until I did research myself in my lab, and I realized that what we were seeing was an embodiment of what Otto Warburg had seen many, many years ago. So I decided to look more deeply into what he said, and I collected data supporting what Otto Warburg said. And then I realized why was there such a negative reaction to Warburg's view. They all refer to his work as the Warburg effect, which is the continual fermentation of producing energy through non-oxidative means, even in the presence of oxygen. Yeah. And then I realized that Warburg and his other associates had made some errors in how they quantified energy. Warburg believed that 1 mole of oxygen consumed was comparable to 7 moles of ATP produced. And then he also assumed that 1 mole of lactic acid produced was comparable to 1 mole of ATP produced. And he was quite rigid about these quantifications. We have now since realized and done our own research to show that these quantifications are inaccurate. And the field said Warburg was wrong because some cancer cells can consume oxygen at the same rate as normal cells. And one of his big protractors was Sidney Winehouse of the National Cancer Institute. And Winehouse said Warburg was wrong because cancer cells have just as good oxygen oxidative phosphorylation as normal cells. So cancer cells and normal cells can generate energy similarly. And they both Warburg and Winehouse, and today most of the entire cancer field still makes the assumption that oxygen consumption is equivalent to energy ATP produced through oxidative phosphorylation. We have now shown that is not the case. Oxygen consumption in cancer cells is used predominantly for reactive oxygen species production, not ATP production. Oxygen in cancer cells can be used for activities other than ATP production. And what neither Warburg nor anyone else at that time knew is that mitochondria in cancer cells can also produce ATP through a fermentation mechanism, which is the new thing that we have a big paper out now that is showing this. So this is very, very confusing for the cancer field and for Warburg and all the others that preceded. So the cancer cell is taking in oxygen and producing energy through the mitochondria, and everybody would assume that's energy through oxidative phosphorylation. But we now realize that the tumor cell will take in large amounts of glutamine, the amino acid, and produce energy through mitochondrial substrate level phosphorylation, which is a fermentation mechanism inside the mitochondria. So this then has created tremendous confusion, making it seem that Warburg was wrong. Now, I have to be honest with you, Warburg had no clue at that time about glutamine fermentation. It's amino acid fermentation. So we have kind of cleared up the mess associated with the confusion about Warburg's ideas and views. So once we realize that all cancer cells, all major cancers, have OXPHOS inefficiency, their oxidative energy through oxidative phosphorylation is inefficient, but they make up that inefficiency through both, yes, glycolysis, as Warburg said, but it's not. I'll explain. It's not that accurate. And through glutamine fermentation. So it's a glucose fermentation and a glutamine fermentation that will compensate for the oxidative phosphorylation deficiency. And that's essentially what Warburg said at the very beginning. He said you have a chronic damage to oxidative phosphorylation coupled with a protracted upregulation of fermentation, which is true. The problem is he did not know that you could also ferment an amino acid called glutamine. That was not clear to him. So yes, glucose is fermented through the lactic acid fermentation, and glutamine is fermented. It's called succinic acid fermentation. So the cancer cells are dumping out waste products of fermentation, which is lactic acid on the one hand, like Warburg always knew and the field knows, but it's also succinic acid, which we now recognize the cancer cells are dumping that out, and that's a waste product of the glutaminolysis pathway. And those are the pathways that are driving the dysregulated growth of all major tumors. They're driving the dysregulated growth through fermentation metabolism in the cytoplasm, which is the glycolytic pathway, the Embden-Meyerhoff-Parnas pathway, and through the glutaminolysis pathway in the mitochondria. It's called mitochondrial substrate level phosphorylation, and we determined this with my good friend and colleague Christos Xenopoulos at Semmelweis University in Budapest. So the two of us have refigured out the whole cancer problem on how cancer cells generate energy. And Warburg had said this a long time ago. He was essentially correct in the concept. His readouts for quantification of energy were not accurate, and we have now shown the inaccuracy. But his overall view was correct. Now, of course, when Warburg was doing all this work in the 1920s, 30s, 40s, and 50s, he was providing strong evidence that cancer was a metabolic disorder with mitochondrial abnormality. But the entire field ran off chasing genes. Yes, there are genetic abnormalities in tumor cells for sure, but these are secondary downstream effects of mitochondrial damage. So today, if you go anywhere in the world, you'll say cancer is a genetic disease caused by mutations in the nucleus leading to dysregulated cell growth. This is no longer correct. We have clearly shown that those mutations, we and others, have shown that the mutations are downstream effects of the damage to the oxidative phosphorylation in the tumor cell. So the tumor cell produces reactive oxygen species, ROS, radicals. They are carcinogenic and mutagenic. So a lot of the genetic changes and damage we see in the tumor nucleus are not the cause of dysregulated cell growth. They're the effect of the shift from oxidative phosphorylation to substrate level phosphorylation. So we just are reinterpreting all of the observations that have been collected over the last 100 years on cancer, just reinterpreting these observations. Not that the observations are incorrect. It's the interpretation of the observations that requires a reevaluation. So once you reevaluate the data, and right now the somatic mutation theory is the dominant explanation for the origin of cancer. And we have now shown that some cancers have no mutations, which is inconsistent with that theory. We now know that these driver mutations that the field thinks are important are now present in normal cells of our body that never form tumors or neoplastic growth. And we also know from the nuclear mitochondrial transfer experiments where the nuclei of tumor cells are placed into fresh cytoplasm with new mitochondria, you get suppression of growth. And if you take the normal cell and put it in a tumor cytoplasm, you get dysregulated growth. These findings completely rule out a nuclear-driven direction of cancer. So it excludes you. So you no longer can believe that mutations driving the dysregulated growth. What's driving the dysregulated growth is, just as Warburg said, it's an insufficiency of oxidative phosphorylation coupled to replacement with fermentation. And you also have to realize that the mitochondria control the differentiated state of the cell, the quiescent differentiated state. And when that organelle becomes chronically disrupted, the cell falls back on its default state, which is proliferation driven by a fermentation metabolism. We now can put all of the facts of cancer in their rightful order for the first time and reevaluate the nature of this disease, which now becomes a mitochondrial metabolic disorder driven by an abnormal fermentation metabolism. So once you understand this, and I know I have dozens of papers written on this subject, goes into great detail about what I'm saying. But now, if we know that the tumor cells cannot, they have a bad mitochondria, defective mitochondria, so they can't use fatty acids or ketone bodies as an alternative fuel to glucose. Respiration, let's put it that way. They can't respire fatty acids or ketone bodies. So that eliminates their ability to be flexible. So the solution, in our view, to the cancer problem is transitioning the entire body over to fatty acids and ketones and put the patient into nutritional ketosis, and then simultaneously restrict the availability of glucose and glutamine, the two fuels driving the fermentation engine. So once you do that, then the tumor cells will be marginalized and killed as the result of this transition. So you transition the whole body over to ketosis, and then lower glucose, which will lower glucose naturally by dietary means, and then you come in with drugs that target the availability of glutamine, and these drugs become less toxic than the drugs that we're presently using. And we developed the press pulse therapeutic strategy on how to use this new understanding to simultaneously kill tumor cells while enhancing the health and vitality of normal cells of the body. So it's a gradual degradation of the tumor by starving it of its fermentable fuels and enhancing non-fermentable fuels for the rest of the body so they can remain healthy while we're degrading the tumor. So what I said to you is so much information put into a little nutshell, but supported by massive amounts of scientific evidence that we and others have collected.
How would you describe this in simple terms for the general public to understand?
I thought that was simple.
Not sure.
that was the simple terms, my friend. You want the more difficult terms, you read the papers that we wrote. Then you'll come to realize what it involves. No, it's very simple. It's not complicated. If you do water-only fasting or restricted ketogenic diets, your blood glucose goes down and your blood ketones go up. This is an evolutionarily conserved adaptation. We as a species evolved to starve. We were always in a semi-ketotic state because we did not have highly processed foods in our environment. We were always active. We always had a very great amount of activity. Our blood sugars were low. Our insulin sensitivity was high. And we were predominantly burning fatty acids and ketone bodies on a regular basis. And that's who we were. Over the last 7,500 years, we have become more and more sedentary, and we have become more dependent on highly processed foods. We've under a lot more emotional stress. We have a lot of things, and all of these impact negatively on the mitochondria function, putting us at risk for a variety of chronic diseases, cancer being a major one, but type 2 diabetes, obesity, Alzheimer's disease, cardiovascular disease, all of these different kinds of diseases are the result of our transition from a very active, nutritionally, ketotically dependent species to a much less active, more stressed, and more dependent on foods that were not part of our evolutionary past. So all of the members of Western societies are now becoming more at risk for chronic diseases as the result of our science and technology advances. But as I said, we are still a Paleolithic man. We evolved over hundreds of thousands of years as hunter-gatherers, and only within the Neolithic period began at about 10 or 12,000 years ago, when we started to grow grains, and civilization built around food sources. We didn't have to be killing and hunting animals. We were able to put domestic animals and plants, rice, wheat, and things like this. They provided greater amounts of quick energy, for sure, but only within the last 70 years have we really exploited these capabilities, producing high fructose corn syrup, poorly nutritious foods, putting us all at risk for these chronic diseases, which then damage oxidative phosphorylation, causing a transition to fermentation, dysregulated cell growth, and cancer. So once you understand everything, it's really not that complicated.
Does this go for all cancer types?
Yeah, so I did a big study on this. Warburg clearly said it was all cancer, was dependent on this metabolism, this abnormal metabolism. And I did a study where I went back and I looked at all major cancers, the ones that representing 95 to 98% of all cancers. And I looked at the previous studies of electron micrographs of mitochondria. And I was clearly able to show that all major cancers have abnormalities in the number, structure, and function of mitochondria. All major cancers accumulate lipid droplets in the cytoplasm, which is a biomarker for abnormal oxidative phosphorylation. So all the parts of the puzzle now make complete sense. So all major cancers have the same metabolic problem, regardless of the organ from which they arise, whether it's a breast tumor, a colon tumor, prostate cancer, bladder cancer, brain cancer, lung cancer, all of these different kinds, blood cancers, the leukemias. They all have the same problem. They're all dependent on a fermentation metabolism because their oxidative phosphorylation system is inefficient. They have inefficient OXPHOS, thereby falling back on a fermentation, leading to dysregulated cell growth because the organelle that controls cell growth is defective and it's no longer in control of the system.
Yeah. What about children's cancer?
Yeah, well, the same thing. When we look at children's cancers, they all are fermenters. So what happened during the development of the child in the utero, in womb, or shortly after birth, that would lead to damage to oxidative phosphorylation in a particular population of cells? Like, for example, the childhood high-grade glioma is the number one killer of children, the number one cancer killer of kids. Every high-grade glioma that we looked at is they're fermenting, so they have that same problem. How did that happen in these little kids? And the answer is we don't know. It could have been a variety. It could have been chemicals. It could have been some developmental abnormality in the growth of that child, causing a chronic inflammation in some part, in some tissue. All we know is that when the tumor arises, whether in a child or an adult, they have damaged oxidative phosphorylation and are dependent on a substrate level phosphorylation. So now we know how to manage childhood cancers as well as adult cancers.
How are you treated, or should I say greeted, by the medical establishment?
Well, I would say, like all paradigm changes, you're ignored. You're just not. It's not that you're not taken seriously. You're just ignored, because you have to realize that most of the funding for cancer, whether it's through federal governments, whether it's your government, the U.S. government, British, German, doesn't make any difference. They all say cancer is a genetic disease. And in order to incorporate what we're saying, you have to assume that gene mutations are causing the oxidative phosphorylation defect. And we have shown that it's just the opposite for somatic mutations. It's the abnormal mitochondria causing the gene defects, not the reverse. So then people come after you and say, whoa, what about the germline mutations? What about, Li-Fraumeni and retinoblastoma and BRCA1 and breast cancer and all these? So we did a big dive on those, and we found out that all germline mutations inherited from one generation to the next, all of these mutations damage oxidative phosphorylation in the same way. So the mutation is a secondary risk. The mutation is a risk factor for damaging oxidative phosphorylation, leading to substrate level phosphorylation, which is the fermentation. So we went through and we did all that. And we know that they're not primary causes of cancer because there's no germline mutation that's 100% penetrant, meaning that everybody that has that mutation develops the phenotype. No, these can vary tremendously. The most penetrant is the Li-Fraumeni, which is a inherited mutation in the P53 gene, which is about 90% penetrant, but still not 100%, meaning that it's a secondary risk factor. All others are much lower. BRCA1 is about 50%, the breast cancer one, retinoblastoma maybe 40 or 50%. All of these are less than 100%, meaning that they cannot be primary causes, they're secondary causes. But every one of those cancers has defects in mitochondrial number, structure, and function. So clearly that's the primary cause. The primary cause of cancer is chronic damage to mitochondria, coupled to a compensatory fermentation, or as we refer to it as substrate level phosphorylation. This is cancer. This is what cancer is. And it happens in all cancers that we know of. We haven't found one. We have yet to find a cancer that does not follow these observations.
Okay. How about the role of carcinogenics since the Industrial Revolution, for example? I'm not speaking about in food. I'm speaking about in general that you're exposed to.
Yeah, well, this is another secondary risk factor, carcinogens of the environment. You can have a variety of different. That's why we have all these chemicals called carcinogens. Why they call a chemical a carcinogen? Because it has the potential to cause cancer. That's why they call it a carcinogen. Okay. And what I've done, aflatoxins and tetrahydrochloride and all these different things, I found that they all chronically damage oxidative phosphorylation, leading to secondary fermentation and dysregulated cell growth. So I have linked all of the known risk factors for cancer, whether it's chronic inflammation, radiation, carcinogens, inherited genes, age, viruses. Let's talk about the papilloma, hepatitis viruses. All of these oncogenic viruses all damage oxidative phosphorylation, leading to transition to a fermentation substrate level phosphorylation. They're all linked to the same thing. So all cancers are essentially the same. They're all an example of a shift from one form of energy to another form of energy. And as Warburg said, if you can't shift from oxphos to fermentation, you can never become a cancer cell. So, and if you are acutely damaging oxphos, you trigger the apoptotic signaling system and you have programmed cell death. So only cells that can compensate for chronic damage to oxphos with a compensatory fermentation, only those cells can become cancer cells. And an example of this is we rarely find tumors of cardiac muscle or neurons of the brain. These kinds of cells, if they sustain damage to oxidative phosphorylation, they die, and you can never get a cancer cell from a dead cell. So you have to have the two components together: chronic damage to oxidative phosphorylation with a protracted upregulation of substrate level phosphorylation. And neurons and cardiac myocytes cannot do that. And that's why you rarely find those cells to form a neoplastic tumor. So everything makes sense. We can account for almost every phenomenon of cancer can now be accounted for when we look at cancer as a mitochondrial metabolic disorder.
Yeah. So let's say I get diagnosed with cancer today. What do I do?
Well, let's say I got diagnosed with cancer today. What would I do?
Yeah, that's a better question.
Right? Okay, the first thing I would do is I would transition my body to a very, very low zero carbohydrate diet. Okay. Number one, I know we have the glucose ketone index monitor. I developed that. We developed that at Boston College. It lets us know when we enter into the state of nutritional therapeutic ketosis. So we have low glucose and we have elevated ketones. That's the first step. What that will do is reduce inflammation of the tumor. It'll reduce the blood vasculature of the tumor. It'll make an angry situation less angry. However, it's not likely to remove the tumor completely. You'll kill a lot of tumor cells doing that, but you may not kill all of them. So what I would do then is after 10 to 12 to 14 days of zero carb, I would then jump into a water-only fasting because the transition now is much easier. Your body has already acclimated to this. And then I would take the drugs that I know will target the glucose and the glutamine availability of the tumor, which is deoxynorlucine, a powerful glutamine targeting drug. And I would take embendazole or fenbendazole and some other parasite medications because I have found that the parasites and the tumors use a common energy pathway to stay alive. So we know we can use those, and they're pretty much non-toxic. There would be very little, if any, toxicity associated with this. And I would have my friends pulse the dawn into my body while I'm in nutritional ketosis. And I would monitor the tumor using non-invasive technology, which is MRIs or PET scans, let's put it there, MRI, CAT scans. No biopsy. I would never do a biopsy. Puts me at risk for spreading the tumor around my body. So there's a lot of things and then I would just try to manage it for as long as I could using these non-invasive imaging techniques and see how I would survive. We've seen it work on a number of other folks. So why wouldn't it work on me if I did it? Now, would I like doing this? No. Is it prohibitively difficult? No. I mean, it's inconvenient. It's not easy to give up carbohydrates. And I would do a lot of exercise. I would do a lot of walking, cardiovascular exercise, which lowers glucose and ketones, while I'm in this new nutritional state using specific drugs, according to what we wrote was the Press Pulse therapeutic strategy for managing cancer, which will eventually become the standard of care at some point once the oncology field comes to recognize what I'm saying. And it will take time. It will take time because most of the oncologists in the world have never heard of what I just said. They were never trained in the medical schools to know this. So, but the people will know, and the people can do it. They just need trained physicians to work with them to do this. And even though I know what to do, and I've written half the stuff about this, I would still reach out to some of my friends who are working daily with cancer patients, and I would talk to them about it. And I would do all these kinds of things mentioned in our Press Pulse metabolic therapy strategy, and hope for the best. But I would not subject my body. Now, I could take some chemo, but it would be very low dosages, because once you're in nutritional ketosis, some of these chemotherapies can be very powerful at very, very low dosages. Unbelievable. Radiation, I would consider that for some part of my body, but never the brain. I would never have my brain irradiated, that's for sure. But there might be a small dose of radiation that could possibly kill off a few surviving tumor cells while minimizing toxicity to the rest of my body. And same with immunotherapies, even though they're based on the somatic mutation theory. I mean, if you can shrink a tumor down to just a few surviving cells, they may all have something in common. They may have a marker on their surface that make them all similar, and therefore an immune therapy could potentially eliminate them. So it would be a stage process, seeing how the tumor would respond to these different approaches as I would go through the process. Maybe I would need this, maybe I would need that, but maybe I wouldn't. So it all depends on the visualization of the tumor using non-invasive technologies while keeping my body at maximum physiological health.
Yeah.
You got to keep your body. You don't want to take anything into your body that has a remote possibility of harming you. This is the key thing. The body is a powerful healing machine if given the opportunity to heal. And you want to enhance the healing capabilities of your body, not taking in toxic chemicals, drugs, or procedures that would harm the ability of your body to heal itself. So again, this whole strategy of metabolic therapy involves natural healing, non-toxic drugs, and a press pulse strategy, as we published. All the stuff that I'm saying right now to you has already been published. So it's just a matter of people knowing what to do and how to do it.
Has there been significant progress in the traditional cancer treatments the last, let's say, 50 years?
Yeah, well, I'm saying we have millions and millions of cancer survivors being treated with toxic poisons, mutilation, and all this kind of stuff. But they pay a significant price for this. Many of these folks, they all of a sudden develop other kinds of problems, right? I mean, if you're being treated with doxorubicin, the Red Devil, or these, you're harming yourself. You're damaging mitochondria, you're damaging DNA, you're damaging the health and vitality of your normal system. A lot of times you get neuropsychiatric problems, hormonal problems, bone density problems as the result of surviving these horrifically toxic procedures used to manage cancer. Why can't we manage? Metabolic therapy will allow us to manage the tumor without producing all these other toxicities. So yes, millions of cancer survivors, but many of those people pay a significant price in overall health, to their overall health and vitality as the result of being subjected. And all of that will increase the second law of thermodynamics, which is entropy. Entropy is disorder. So when your body is damaged, you have disorder, and that will reduce your survival on the planet for most people, not all people, but certainly. So I try to avoid all that stuff. Why would I do that if I didn't have to do that? So, this is the strategy. This will be the future. It's just that it will take time for the people to understand what I'm talking about.
Which role does exercise play?
Yeah, very important. As I said, you can lower glucose and glutamine by exercising. So clearly your mitochondria get healthier and you're depriving the tumor of the very fuels that it needs to survive. So exercise under nutritional ketosis is really powerful. So will really enhance the health and vitality of your normal cells while putting tremendous stress on the tumor cells. Absolutely. That's part of the press pulse therapy, exercise.
Okay. So, for example, exercising under fasting would be beneficial.
Oh, yeah. Is it easy? No. But you have to realize our ancestors did that all the time. They wouldn't be trying to run down and track down big and kill animals if they could go to a delicatessen and get a big sandwich. They did that because they had to do hundreds of thousands of years. So that was exercise under nutritional ketosis. And then, of course, when you make the kill of the animal, then you can eat as much food. But you have to realize that food has very little highly processed it has no highly processed carbohydrates in that. So yeah, our ancestors were very active, and they were mostly in a semi-nutritional ketotic state because of there was no highly processed carbs in the environment, and they had to be active, otherwise they'd starve to death. So we're just simply going back to our evolutionary past.
Yeah. What about fruit? There's a lot of glucose in fruit.
Yeah, well, fruits in our past were seasonal. And also the fruits that we had in our past were not genetically engineered to be full of sugar. So you had to wait for the fruit to become ripe at a certain short period of time in our existence, and then you would eat the fruit as much as you possibly could just to get that sweetness out of the fruit, and it would be like a little delicacy. But today we have fruit all the time, all seasons, and they're genetically engineered to be quite sweet. So you can taste it in the fruit. There's a lot of fructose in the fruit. Not that it's so bad. It's just that you use the glucose ketone index calculator and then eat an apple or an orange today and see if it throws you out of ketosis. Because if it does, then you might want to restrict. What's good that we learned when I was working with the epilepsy for years with ketogenic metabolic therapy for epilepsy? Grapefruits. Grapefruits have a lot of vitamin C, are very healthy, and they have very low glycemic index. So because in children with epilepsy, if they're managed quite effectively on ketogenic metabolic therapy and they were to eat one little sweet thing, they would blow into a seizure, a breakthrough epileptic seizure. So my colleagues realized that grapefruits can provide very significant vitamin C without spiking the sugar, and therefore you could maintain a lower GKI without causing a problem. So there are some fruits for sure that can do this, and grapefruit is one of them.
Have you received any trouble in getting funds for your research?
Yes, that's a real problem. That's, as you can imagine, right?
Yeah.
So, but we got some nice funding from private foundations, and some of these private foundations, they're interested in outcome and results. If you have a childhood brain cancer, we got a very nice support from the UK, United Kingdom Childhood Cancer Fund, and we have funding from Travis Christofferson's foundation for cancer metabolic therapies, and some of our most important funding has come from philanthropy. To both my university and to Travis Christofferson's foundation. There are people on this planet that want to be part of this paradigm change, and they don't care whether they make money on what we're going to do. They just want to say that this will be the future for cancer management, and we'd like to support your research because we are spearheading this whole thing. Our preclinical studies that I do in my lab serve as the platform for translational to the human. So everything that I have told you so far comes from our preclinical studies and observing how that strategy works for people. And we see that the human being responds in a much more favorable way than some of our preclinical studies, our preclinical mouse models of these very aggressive metastatic brain cancers and other kinds of malignant cancers. But we understand how to use and manipulate the power of diets and drugs. So in these preclinical studies, so that when the clinician goes and treats his patients, he will know already the things that work best, and then he will modify those and perfect those as he goes through his patient population. So our preclinical studies are absolutely crucial for the translational benefit to humans. So as long as we can continue to work out dosage, timing, and scheduling, looking at the key drugs and diets that work together for this, then we immediately translate that to the clinic, and that's where we're going to see the big changes. But that comes from the funding that I get. Yeah, because it's hard to get money from the federal governments when they think cancer is a genetic disease. So why are you funding metabolic therapy when cancer is a genetic disease? But people who want to see results and they want to see loved ones saved and they want to keep themselves alive longer at a healthier state, they're the ones who would want to fund us, because the results will be helping them directly. And that's what I do. I give kits of information to the thousands of cancer patients who contact me. And then I ask them, if this information helps you, consider making a donation to the Travis's foundation or to the university that supports my research, Boston College. And they do that. And we're very thankful. And not only that, when they make these contributions in the papers that we publish, we acknowledge them in the acknowledgement section. So when people look at my papers and they say, oh, go to the acknowledgement section, they say, oh, look, who are all these people? Who are all these things? These are the guys who are supporting our research. So they feel good that I acknowledge them in a scientific publication, that they're contributing to the success of what we're doing.
What about you said it there, but what about the college? No problem with doing this research at the college.
Oh no! I mean, this is we're doing basic research. I teach a class in cancer metabolism. I mean, all this is integrated with cell and molecular biology that the department focuses on. It's just that we're focusing on the cancer problem rather than on some other problems that that diabetes and these kinds of things. But the college, no, the college is very supportive of what we do.
What are the next steps, as you see it, to raise more awareness about this?
Well, continuing to publish the papers and the scientific evidence that supports what I'm saying. So this concept of mitochondrial substrate level phosphorylation using glutamine is a new concept in cancer. And we have to test that in all major forms of cancer. We have to know that all breast cancer, colon cancer, lung cancer, brain cancer, all of the different cancers, no matter what you look at, are using a similar pathway to generate energy. We've proven that in some brain cancers. We have strong evidence in brain cancer. We need to show that in lung and the other cancers. But I know it will be shown because the cancer cells already tell us what their problem is. When we see lactic acid and succinic acid produced, they are telling us that they are fermenting glucose and glutamine. We just have to show the pathway. We just have to prove that in designed experiments. And we're also working on some novel diet drug combinations that are potentially patentable, which would then lead pharmaceutical companies to be more excited about using metabolic therapies to treat cancer. So we're working on that as well, because we have to realize there's a multi-billion-dollar industry around cancer, and we have to be able to say that there are certain strategies that can be very, very profitable if proven to be correct, and we're doing that. So this is what we're doing right now.
So how much of the problem do you think is because of companies making a lot of money on the traditional treatment versus getting the information to the right people?
Yeah, well, this is one of the biggest problems, because you right now you have a very successful revenue generation system for managing cancer. These immunotherapies, CAR T immunotherapy, all of these very toxic drugs, radiation, they're all very big revenue generating systems. So how can you move in a new concept and a new design without disrupting too terribly the revenue streams? And that's what has to be integrated. So in all likelihood, we'll have some sort of a hybrid system coming together, metabolic therapy with standards of care, and eventually modifying standards of care to recognize more of the metabolic approach. So it's going to be a gradual transition. But the bottom line is, it's not something that we should drag our feet on, because right now in the United States, we have 1,700 people a day dying from cancer. That comes to about 70 an hour. And we don't want to be dragging our feet too long on this when we can keep a lot of these folks alive. So and it goes for all cancer, all throughout the world. I mean, there's people dying everywhere from cancer. We have a clear strategy for managing this disease without toxicity. So, and we want to make the transition over so everybody can benefit. That is, the pharmaceutical companies, hospitals, as well as the cancer patients. So that requires people who understand that part of the strategy. Unfortunately, I'm not an entrepreneur. I'm the one who designs the framework and says this is what we have to do based on the hard science. And there'll be other really bright young people that'll come in to know how to make this transition acceptable to the majority of people. But we have to wait for that. But as I said, we don't want to wait too long, because this is not an insignificant problem.
No. I imagine you've spoken to several doctors about this. What did they say?
Some are extremely important and absolutely understand what I'm saying based on their long history of treating cancer patients. Others are very resistant because they have never been trained to understand what I've said, and they feel sometimes embarrassed and threatened, not knowing what's happening. But I think that in time, and that's another thing, we have to start training oncologists to know about what I'm saying. They're trained to know how to use radiation and chemo and these other kinds of things, but they're not recognizing that cancer is a metabolic problem. How do we use diets and drugs to achieve a similar outcome? Why are we doing what we're doing? What's in the best interest of the patient? How can we blend certain of these procedures in together? So it's going to be a re-education of the medical field in this area of medicine.
Yeah. You opened by saying that Otto Warburg said all disease is metabolic.
No, cancer. But
Okay.
he wasn't dealing with obesity. He wasn't dealing so much with type 2 diabetes. But we can link the obesity epidemic to cancer. We know that obesity in many people produces systemic inflammation.
Yeah.
Systemic inflammation leads to chronic mitochondrial damage, leading to dysregulated cell growth in a particular populations of cell in somebody's body. So type 2 diabetes is linked to obesity. Type 2 diabetes and obesity are linked to cancer, cardiovascular disease, even dementia. Many dementias are the result of metabolic abnormalities. So, our ancestors, I don't know if they've lived long enough to get certain kinds of dementia, but we know that dementia has increased significantly over the last 50 years compared to what it was. All these chronic diseases have exploded over the last 50 to 100 years.
What about Alzheimer's?
Well, there's 5 to 8% of Alzheimer's are due to serious mutations, presenilins and things like this, and you really can't do too much about that. But a good 40 to 50% of dementias are linked to metabolic problems. So, if you burn ketones, you're going to protect the mitochondria, and you're going to reduce inflammation in the brain, which is associated with the plaques and tangles. So, all of these things are linked together in certain ways.
Is there anything else in particular you would like to mention about your research?
As long as we have funding, we can continue to make these developments. I collaborate with Dr. Shinopoulos at Semmelweis University, and we're beginning collaborations with clinics. And I think that the solution will eventually be our work on preclinical designs, preclinical testing, and translation to the clinic. So and they tell us from the clinical findings in humans what seems to be really, really effective, and then we go back and modify the preclinical system in a way that we can enhance the therapeutic efficacy in the preclinical system based on some of the information we get in the clinics, and then we even perfect it more, and then it goes back to the clinic. So you have to realize we're at the very beginning of this whole thing. The science is now established. The practicality and implementation now have to be perfected. And that's dosage, timing, and scheduling of these different procedures according to the press pulse therapeutic strategy for the metabolic management of cancer. So we have a lot of the framework. We just have to flesh it out now and see what works best for various people at different ages. We have to recognize that, like, what works, how to treat a child, a young child versus an old, a senior citizen. We have to be cognizant of dietary and cultural differences among people, religious differences that restrict certain foods and things like this. So all of this will be blended and developed as we go through the system.
So in summary, which advice would you give to people who are today healthy and want to stay that way?
Well, that's probably the most important thing we can do. So if we understand that all cancers arise in one way or another from chronic damage to oxidative phosphorylation, which is the health and vitality of your mitochondria in your body, in cells, then protecting the mitochondria from that kind of damage will go far in preventing cancer in the first place. So, that our society has an obesity epidemic clearly indicates that people may not really be concerned or care about maintaining mitochondrial health. I mean, that's a statement. I mean, if people were really, really interested in what I was saying, you would rarely find an obese person walking around anywhere. So clearly they either don't know or don't care. But to prevent cancer, you really have to keep your mitochondria healthy. And, it's interesting. Not all obese people have systemic inflammation. There are some people that are quite obese, but they're very, very healthy. But the majority they're the outliers.
Why?
We're not really sure about that. They have great insulin sensitivity, and they have low inflammation, and they would be considered obese. But we have many other people who are obese, and they have chronic inflammation, and they have the markers of unhealth. They have high triglycerides. They have chronic C-reactive protein, whereas some obese people don't. But it's usually it's not common to see that, but it can happen. And the answer is, I don't know. Something about their genetic makeup or something that allows them. You have to realize that the obese people that we see today are essentially the descendants of our Paleolithic ancestors, because as a species, we survived to be able to go long periods of time without food. And we were supremely designed to store energy in our body as effectively as possible to allow us to hunt animals and move great distances in search of food. So these are the descendants of ancestors that were supremely capable of storing energy. Now we put our species in an environment where highly processed carbs and energy storage can happen so easily. So we have this. It's just our biology is still Paleolithic, whereas our science and technology has moved so much faster than our biology, and we're just coming to realize that, and how it's putting us at risk for all these chronic diseases. So once we coordinate our biology with our technology, we will move into a new healthy state where we can enjoy all of the things that our technology has provided without abusing them and overdoing it. And that way we can still enjoy the greatest things that we've developed without putting ourselves at a tremendous risk for chronic disease. It'll be really, really, really interesting to see. And it's also the knowledge base. People will have to become scientifically literate, knowing about some of these things, understanding how your body works. And that's one of the things I do here at Boston College. I spend a lot of time with my students talking about scientific literacy. They need to know about these concepts, so it improves their overall quality of life.
What about smoking and lung cancer?
Well, of course, in the 1990s, the United States embarked on an anti-smoking campaign because secondhand smoke was affecting others. So it became socially unacceptable by peer pressure to reduce smoking. So they used to have non-smoking and smoking sections of restaurants and buildings. Then all of a sudden it went to completely non-smoking, and now you have to go outside somewhere to smoke. But that has reduced cancer deaths by about 30%. So when the NIH, the National Cancer Institute, says we've made tremendous progress in reducing cancer deaths, I would say 90% of that progress has due to the anti-smoking campaign, which is basically a prevention campaign, not due to any new drugs or procedures, but rather to the people's behavioral change. So, the obesity epidemic is allowed to continue because people feel that, one person's obesity is not going to have an effect on my health. as secondhand smoke would have if someone were to be smoking. So there's no peer pressure to reduce the obesity epidemic, which is linked to type 2 diabetes and chronic inflammation, because that's pretty much within that person's own self. So, so you don't have as much peer pressure to reduce that. As a matter of fact, the commercials are now gravitating more and more to fashions that fit obese people, diets. I mean, it's just we're not doing the right thing. We're going in the wrong direction.
Last question about this. I had a grandfather who I think he smoked a pack or two for 75 years of his life and lived to be 90 and never sick. Is that kind of the same mechanisms that you spoke about in some obese people being healthy still?
Yeah. So these guys are what we call outliers. You know, the greatest outlier of all time. I use this. To me, the greatest outlier of all time is the Japanese fellow named Yamaguchi. Okay, he's one of the few, if not the only person to survive 2 direct hits from atomic bombs on Nagasaki and Hiroshima. Direct hits. He survived both. He was active. He lived to be 92 or 3 years old, having survived. He's called an outlier. All right, yeah. Not many people. So the guy who smokes 6 packs of cigarettes a day and lives to be 95. You can call him an outlier like a Yamaguchi. But most of the people that do this don't survive. They all die at a younger age. Yes, there are always an outlier. And people say, you see that guy over there? You know, he was morbidly obese. He smoked, he drank, he did everything. He lived to be 85 years old, 90 years old. See, I can do that. No. He's what they call outliers. He's not the norm. He's the very exception. So yes, we do have outliers, and a lot of people look to them and say, see, it's not so bad. But if you look at the general overall statistics, you generally don't survive that long.
No. What do you think of the cold plunge, sauna use, and all these breathing techniques, etc.?
Yeah, well, I did that. It's no fun. You know, I don't. I mean, you have to be. I did it up in Canada. The plunge. Oh yeah, it's good. Tell me how good it is. Are you kidding me? Who the hell wants to jump in ice cold water? Give me a break. You know. Hey, but we always found an outlier out there. You always see these guys, right? I mean, this guy Nip from Germany take a bath in the middle of the Rhine River in the middle of January. You know, these guys. You know, some guy standing in a cold shower screaming while the cold water is on his back. You know, I don't know. I mean, it makes that guy feel good. Hey, my hat's off to him. I'm not that kind. And I'm not going to be. I didn't enjoy it. Let's put it this way. It wasn't something top on my list that I would do. So I'm not saying that it's not going to be healthy for you. No, I'm not saying that at all. But I'm not that kind. I'd rather do the other things that I know what I can do.
What about red light therapy?
I have no idea. You know, I've never tested that stuff. A lot of this stuff is like, just, you know, it makes you think something, but it's show me where it's targeting glucose and glutamine. You want to kill cancer cells, you got to target glucose and glutamine. If red lights can do that, you show me the scientific evidence, and maybe I'll look into it.
Yeah. Last question. What's the meaning of life?
Avoiding entropy, second law of thermodynamics. We're all going to die. It's just a matter of time, and that's based on the law of thermodynamics, entropy disorder. So if you can do something to delay entropy, you might live longer. Because don't forget, when you no longer have an electric charge on either side of your membrane, that's called equilibrium, and that's called death. And some people think that's heaven. When you're in equilibrium, you no longer have any anxieties. You don't have any pressure. You don't have anything. You're at equilibrium. Some people would call that heaven. Yeah. Chew on that for a while.
Thank you very much for your time.
Okay, Alexander. It was very nice speaking with you.
You too. Tusen takk for at du hørte på Bendiksen Airways. Dersom du har tips, kommentarer eller vil gi en vurdering av episoden, så gjør gjerne det. Det setter jeg stor pris på.